Systems-wide studies are crucial for the in-depth understanding of the biological functions. However, multiple independent samples are required for different omics platforms, introducing high amount of variability to the data. This method offers a robust and high throughput strategy for simultaneous extraction of chlorophyll, lipids, metabolites, proteins, and starch from a single sample of the model green alga, Chlamydomonas reinhardtii.
For chlorophyll, lipid, and metabolite extraction, arrange the tubes of harvested Chlamydomonas cell pellets in liquid nitrogen. Resuspend the pellet in each tube with 1 milliliter of -20° extraction buffer 1. To avoid evaporation of the low-viscosity extraction buffer, quickly vortex the tubes until the cells are well homogenized within the extraction mixture, and aliquot the solution into 2 milliliter microcentrifuge tubes. Sonicate the cultures in a sonication bath in ice-cooled water for 10 minutes, before incubation on an orbital shaker at 1000 rotations per minute for 60 minutes at 4 degrees Celsius. At the end of the incubation, add 650 microliters of extraction buffer 2, and briefly vortex the samples before centrifugation. To aliquot the fractions, transfer 500 microliters of the upper MTBE lipid phase into a labeled 1.5 milliliter tube. Use a 200 microliter pipette to remove the lipid phase, and transfer 650 microliters of the lower polar and semipolar metabolite phase into new labeled tubes. Aspirate the excess volume to remove the remaining lower phase, and freeze the solid pellets in liquid nitrogen.